Let's start. तो कोई भी नहीं है टाइम है 10:30 हो
Yes, sir.
Start. So this is the third and last lecture in the series, in the lecture series. So in the first lecture we started with the definition of what cancer is. It's an uncontrolled, not cancer, tumor. It's an uncontrolled mass of cells or uncontrolled growth of cells. Sometimes these tumorous outgrowths they are changed into cancers when they become malignant. Malignant means they spread to other bodily regions as well. Then we move on to genes that are important for cancer development. The first is the oncogenes, which are basically first proto-oncogenes and then later they become oncogenes when they are directly linked with the emergence of tumors and cancers. Some examples are, क्या examples है? कौन सी है? Oncogenes की BRCA1, BRCA2 और कौन सा है? Any other? MYC, RB genes हैं, जीन रॉस, वो तो सेकेंड कैटेगरी हो गई ना. फर्स्ट कैटेगरी है oncogenes की एग्जांपल्स हैं BRCA1, BRCA2, RAS, SARC. These are the examples of oncogenes जिनमें yes MYC as well, जिनमें कोई भी mutations हो जाए then they can be converted into oncogenes. Normally they are called proto-oncogenes. So उनकी prototypical original form है. Then we started discussing tumor suppressor genes whose functional deactivation may cause the emergence of tumors. जो oncogenes हैं उनमें gain of function, if they somehow gain an additional function to grow the mass of cells, while in tumor suppressor genes or TSGs, their inactivation, which is a recessive expression pattern. Recessive means you need to have the mutated versions of both the alleles. दोनों जो जीन कॉपीज हैं उनमें म्यूटेशन होनी चाहिए. Only then a tumor suppressor gene will lose its functionality, and that loss may culminate into the emergence of tumors or cancers. इसकी prime example जो है वो P53 है. Then in the second lecture we started discussing different pathways like the P53 pathway, which is more related to TSGs, and also pathways that are related to oncogenes as well. So when we talk about the pathway or cascade, then we may be talking about a set of different genes that are functionally linked to one another. बहुत सारे genes होते हैं pathway में जो कि एक दूसरे के साथ functionally linked होते हैं. And mutation in any of the underlying genes may be causally linked to the emergence of tumors or cancers. तो जो भी रिसर्च हो रही है ऑन्कोलॉजी में, particularly cancer genomics में, वो इन्हीं जीन्स के इर्द-गिर्द होंगी. या maybe nowadays because of the NGS they do unbiased sequencing. एक होती है biased sequencing. Biased sequencing means जब आप किसी particular gene के expression या sequence को target करते हैं. Unbiased means you don't target anything, you just take a sample, throw it into the sequencer, and the sequencer will give you some reads. You will process those reads and see what genes are activated, what genes are being deactivated. So this is an unbiased approach. आप टारगेट नहीं कर रहे, अनटारगेटेड है ये. यहाँ से आपको न्यू प्रोटीन्स और न्यू जीन्स के बारे में भी पता चलता है. कौन से वो न्यू जीन है जो कि एक्टिवेट या डीएक्टिवेट हो रहे हैं। कैसे पता चलेगा? उनके एक्सप्रेशन लेवल से पता चलेगा। You record their expression levels, compare it with the expression of housekeeping genes, and you will see whether they are being deactivated or activated. ये वो तरीका है। इसके अलावा एक्सपेरिमेंटली आप कैंसर सेल लाइन्स ग्रो भी कर सकते हैं and see how they behave against different molecules. Small molecules हैं जिनका molecular weight कम होता है। We use small molecules because they can be easily synthesized in a lab compared to high molecular weight compounds. तो ये भी एक रिसर्च एवेन्यू है जिस पे आप काम कर सकते हैं। आजकल HTS स्टडीज यानी हाई थ्रूपुट स्टडीज हो रही हैं जिसमें आप एक ही वक्त में you actually screen thousands of molecules and see the best hits. उन molecules में से कौन से ऐसे हैं जो कि further फिर आप आगे ले जाकर उनकी कैरेक्टराइजेशन कर सकते हैं। इसके अलावा कैंसर सेल्स को माइक्रोस्कोपिकली भी देखा जाता है। यू मे यूज कंफोकल माइक्रोस्कोप, यू मे चेंज सम पाथवेज। अगर वेंट पाथवे है, BMP पाथवे, these are all pathways related to growth of cells or cell division. You may use certain molecules to either upregulate or downregulate these pathways and then see the behavior of cells in your petri. These are all experimental things you may do in this oncology field. आगे चलकर आप ट्रांसलेशनल स्टडीज भी कर सकते हैं। फिर एक्सपेरिमेंटल स्टडीज आर प्रॉमिसिंग, तो वही चीज़ें आप आगे फिर एनिमल मॉडल्स में और ह्यूमन मॉडल्स में भी ले जा सकते हैं। उन्हीं में से कुछ आज हम ट्रीटमेंट ऑप्शंस जो है वो आज और कल जो है वो डिस्कस करेंगे। ठीक है, एनी क्वेश्चंस? आप में से कोई है जो कैंसर्स पे काम कर रहा है? आप कर रहे हैं, क्या कर रहे हैं आप?
सर हमारा लेकिन प्रोटीन काउंट है, टारगेट प्रोटीन है स्टेरॉइड सल्फेटेज जो कि ब्रेस्ट कैंसर में ओवर एक्सप्रेस होता है जो कि ईस्ट्रोजन मेटाबॉलिज्म।
ठीक है, आपका क्या एम क्या है?
हम लोग तो कंपाउंड स्क्रीन करते हैं तो प्रोटीन प्योरीफाई करके ह्यूमन प्लेसेंटा से, और वहाँ से हम स्टेरॉइड सल्फेटेज को प्योरीफाई करते हैं और फिर हम उसमें कंपाउंड्स को देखते हैं जो उनको इनहिबिट कर रहे हैं। अब PhD में लेकिन ये है कि मुझे अभी कुछ नए और एक्सपेरिमेंट्स डालने ताकि सेल लाइन पर अगर हम करें MCF7 सेल लाइन हो गई है डिफरेंट, तो उसमें डिफरेंट पाथवेज को टारगेट करने के लिए
देखा ये इनकी रिसर्च। ठीक है, सो शी इज एक्चुअली targeting a cancer protein, yeah, which is implicated in cancers, and she is screening different molecules which can inhibit the protein, and now she is trying to check the pathways, the underlying pathways that are being disturbed, जो कि डिस्टर्ब हुए हैं इसी कैंसर प्रोटीन की वजह से, और कैसे हम उनके फंक्शन को रिस्टोर कर दें या इनहिबिट कर दें। पर्सनलाइज्ड थेरेपी में ये हो सकता है कि जो कैंसर सेल्स की पॉपुलेशन है वहाँ पे ग्रोथ को इनहिबिट समहाउ किया जाए in different ways. We will discuss some of them here in this lecture. So this is the third lecture, cancer prevention and treatment. So the first thing we start with is the epidemiology. You are trying to work on a particular type of cancer. The first thing you do is to get some information from your region, how prevalent that cancer is. क्या वो कैंसर उस पॉपुलेशन में एग्जिस्ट भी कर रहा है जहाँ जिस कैंसर पे आप काम करना चाह रहे हैं, उसका प्रेवलेंस रेट क्या है? प्रेवलेंस रेट यानी कितने पेशेंट्स हैं आपके पास, ठीक है, पर 100 ले लेते हैं ताकि एक परसेंटेज आ जाए। So epidemiology reveals that many cases of cancer are preventable. How? So this is a graph, like what it shows is the increase in the number of cancers. This map of the world, so this is a map that shows the rates of cancer increasing, red arrows. So this is a country, and then this is a country as well. So the cancer is increasing here. Somehow ये कैंसर जो कि पहले यहाँ पे रिपोर्ट हुआ था अब यहाँ पे भी रिपोर्ट हो रहा है, और उसके केसेस जो है वो बढ़ते जा रहे हैं और डिक्रीजिंग। सो फ्रॉम हियर टू here. So we have a region here, Africa. This is the North Africa. So a cancer, maybe whatever type of cancer, is started here in Africa, but now it is decreasing here in North America. When specific populations move from one location to another, such observations suggest the importance of environmental factors, including diet, which may be dictating cancer risk. तो populations move करती हैं, और वो population जो कि एक particular region में highly susceptible थी एक particular type of cancer के लिए, जब वो दूसरे region में migrate किया उस population ने, तो cancer का risk काफी कम हो गया. It means something has been changed in the new region, and that change may be the diet, may be the environment. जो first encounters हैं, आप किसी भी नए मुल्क में जाते हैं, environment आप पर effect करती है, आप नए खाने लग जाते हैं. So maybe that is something that is dictating the cancer risk. This is a table showing some estimated effects of environment and lifestyle on cancer in the United States. The table shows both the yearly deaths in the US attributable to each cancer and the estimated percentage of that cancer that could be eliminated through prevention. So smoking, 33% of the cases are linked to smoking. Then we have around 190,000 deaths annually, and 75% reduction can be possible if we somehow educate people and try to prevent smoking habits in the population. Then we have diet, overweight and obesity. 50% is preventable. Lack of exercise, which is 5%, but then 85% of the cases are preventable. Viruses, 100% are preventable. Alcohol, linked to 3%, and then 50% is preventable by changing the habits, by changing the behavior of the susceptible population. इसके अलावा we have UV and ionizing radiation or occupational carcinogens. Occupational यानी जो काम कर रहे हैं, professional, आप किसी chemical factory में काम कर रहे हैं, maybe working with some chemicals that have the risk of developing tumors. तो आप उस chemicals के साथ या तो पूरे gloves वगैरह पहन कर काम करें या वहाँ काम ही न करें. So this is how it is preventable. So carcinogens, the chemical drivers of cancer, are diverse in structure, but present with one shared property. They cause mutations. There is a diverse set of carcinogens, many chemicals that may be the reasons for the development of cancers. They are diverse in structure. They have different chemical structures. They belong to different chemical types. कोई alcohols होंगे, कोई esters होंगे, कोई let's say carbohydrates होंगे, proteins, whatever. They have different chemical structures, but they have one shared property. They are mutable. They can initiate mutations in your genomes. Sensitive assays can detect those cancer-causing agents that damage DNA. We have certain assays that can detect these agents. For example, Ames test, ये बहुत ही traditional test है. It's a classical test. काफी पहले से ये develop हुआ है. A technique to determine mutagenic potential. So this is the experimental blueprint. Okay. So what we have here, we pour a suspected mutagen and then liver extract. This is experimental tube. Culture of his negative salmonella. What they do is they check this mutagen not on a cell line but on a bacterial culture to see how it initiates mutations in the bacterial genome. medium lacking histidine, and then incubation colony of revertant is positive salmonella. So somehow is जो भी आपका सस्पेक्टेड म्यूटेजन है, जो भी आपका सस्पेक्टेड केमिकल है, जो कि आप समझते हैं कि म्यूटेशंस कॉज कर सकता है, वो चेंज कर देता है his negative salmonella population to his positive salmonella population. Because of certain mutations to the bacterial genome, it is mutated because of the chemical. And जो भी आप लोग बैक्टीरियल कल्चर्स के साथ काम कर रहे हैं, you check the cultures by different markers like antibiotic resistance genes or functionality genes. So Ames test is used to determine the mutagenic potential of chemicals in animals. They may be highly carcinogenic or moderately or not carcinogenic at all. So the test was developed by Bruce Ames. So they just named the test against the inventor who actually invented the assay. So what they do is they mix the carcinogen with an activating extract prepared from rat liver cells to mimic the biochemical processing that occurs in an intact animal. कोई भी चीज जब आप लेते हैं, so the first thing that it encounters with is the liver. Actually, liver processes or detoxify the thing you take in. So this is why they mix it with the rat liver cells. The mixture is then added to a culture of specially designed test bacteria and the bacterial mutation rate measured. So the first thing you do is you take the carcinogen, जो भी chemical है आपके पास जिसको आप टेस्ट करना चाहते हैं, उसको मिक्स करेंगे किसके साथ? लिवर सेल्स के साथ. So if you are taking maybe the liver cells from the rat, so it's a rat liver cell line. I told you because liver cells are the ones that actually process all the chemicals naturally. और ये mixture जो है, then you will add to a test bacterial population. आपके पास कोई भी bacterial population हो उसमें आप add कर देंगे, and then you measure the mutation rate. उसका अपना एक तरीका है mutation rate measurement का, ठीक है? उसका एक formula होता है. You either take the whole number of cells and then see the number of cells that survived. वो आप कहीं से भी देख सकते हैं. Just write on ChatGPT how to measure, how to estimate the mutation rate. तो formula आपको बता दें. और वो जो formula के जो variables हैं, those will be calculated experimentally. आप हेमेटोसाइटोमेट्री यूज करते हैं सेल पॉपुलेशन की काउंटिंग के लिए. Most of the compounds scored as mutagenic by this rapid and convenient assay in bacteria also cause mutations or chromosome aberrations when tested on mammalian cells. So whatever you see like a chemical that has mutagenic potential in bacteria will probably have the same effect in animal cells. So different carcinogens cause different types of cancer. For example, vinyl chloride is linked to liver angiosarcoma. Benzene, acute leukemias. Arsenic is linked to skin carcinomas and bladder cancer. Asbestos with mesothelioma or radium osteosarcoma. So the sarcomas are the cancers of the bone. ये terminology भी याद रखें, ठीक है? जो sarcoma होता है, blastoma, sarcoma, carcinoma. So you have to remember these. This is the basic cancer classification. और उसकी grading जो होती है, grade 1, grade 2, grade. अगर आप cancer research में काम करने जा रहे हैं या कर रहे हैं, then you should remember this. Like someone asks you what sarcoma is, and you can promptly say these are the cancers of the bone, or carcinomas are the cancers of the skin. Blastoma, just like glioblastoma, these are the cancers of the glial cells, neurotumors. So these are some of the chemicals that are linked. That this is the known information. Like if you are working with this chemical, there is a hazard. If you don't take care, it may cause some mutations in your genome. and unfortunately those mutations may be the likelihood of different tumors or cancers. These are the risk factors, not necessarily. ज़रूरी नहीं है कि ऐसा हो, but there is a risk involved. So you need to take care. So for example, we have a chemical aflatoxin. अगर aflatoxin जैसे वहाँ हमने ऐसे में पढ़ा, you mix it with the rat liver cells, and the enzyme that is present in the liver cells is the cytochrome P450 enzyme that actually works with any chemical. It reacts with any chemical. So how the carcinogen is activated? A metabolic transformation in the liver catalyzed by a set of intracellular enzymes known as the cytochrome P450 oxidases must activate many chemical carcinogens before they will cause mutations. So the chemicals are actually converted into their active forms by this enzyme. So here, like aflatoxin is converted into aflatoxin 2,3-epoxide, and then it becomes carcinogen. Then it can do all these mutation initiations in your genome. This carcinogen, it binds to the guanine in the DNA. And G's are there. A, T, G, C जो G's है ये आपके genome में होते हैं. So when this aflatoxin is activated, it then binds to the guanine in the DNA, and because of this binding, the chemical structure is now changed. वो chemical structure भी change हो जाता है, and then you have to see what other effects are due to the change in the chemical structure. तो जो लोग structural biology में काम कर रहे हैं या chemistry में काम कर रहे हैं, they usually study the structures. ये structures जब change हो जाते हैं, of course they have certain effects. इसके effects होते हैं उस protein के functionality पे, उस molecule की functionality पे. यही mutations का भी है. So when a mutation happens, a molecule is being replaced by another molecule. A is replaced by T or T is replaced by A, which is a different molecule. और जब वो molecule उस strand का हिस्सा बन जाता है, then it actually change the functionality of the resulting product. जो भी आपकी product बनती है. So these are the three simple steps that can be adopted. Like you can eat well, you can do exercise and stay lean, like reduce the obesity. So this is how like you can simply decrease the rate of cancers in the society. So now we move to another thing, the viruses. We studied the chemicals that are being linked to the emergence of tumors. Now we will study a different type, viruses, which can also contribute to a significant proportion of human cancers. Cancer in humans is not an infectious disease. It doesn't spread. However, a small but significant proportion of human cancers, perhaps 50% in the world or globally, are thought to arise by mechanisms that involve viruses, bacteria, or parasites. For example, uterine cervix is associated with infection with a papillomavirus, while liver cancer is very common in parts of the world, Africa or the Southeast Asia, where hepatitis B viral infections are common. Cervical cancers हैं ठीक, या आपके पास ये liver cancers जो हैं, ये उन इलाकों में ज़्यादा इनकी prevalence है. They are more prevalent in areas that have the high prevalence rate of the underlying viruses as well. For example, जो cervical cancer है, ये papillomaviral infections के साथ linked है, and the liver cancer is more related to the high infection rate of hepatitis B virus. Again, it's not a must. Not necessarily the virus will cause a cancer, but it may change, it may initiate changes in your genome that if not prevented or not corrected, you or any patient may develop tumors or cancers. So how does a virus cause cancer?
Why do some viruses cause cancer?
So my name is Mark Horswell. I'm a research technician here at the Morgridge Institute for Research. I work in the virology lab of Dr. Paul Alquist. Infectious agents, bacterial and viral, are responsible for about 25% of all human cancers. To date, there's about 7 known viruses that cause human cancers. And the 2 that are most known are hepatitis B and C that cause liver cancer, and then human papillomavirus, or HPV, that our lab works on, that causes cervical cancer. The link between viruses and cancer is not so direct or one-to-one. Cancer by itself, in its definition, is uncontrolled cell growth. And this uncontrolled cell growth is a rather broad term for a complex process of the cell going from a normal state to a cancer state. And the infection alone is only one component for cancer. In and of itself does not cause cancer. It's an initial infection that, for instance, HPV, that will express some viral proteins that will mess up with the cell's normal maintenance of things. So the cell's ability to repair itself will be hindered by products from the virus. And given time, over time, when there's sufficient number of these changes, not just one, but several changes over a long time, these changes cause a transformation of the cell, which we've been deemed as cancer. HPV research has been a great success in the last 40 years. Because we found, as a community, that certain cancer, cervical cancer, is caused by a particular virus, HPV. And that not only have we found this, but in this instance too, the community has been able to develop a vaccine. And so if you get this vaccine before you're actually exposed to HPV, this will greatly reduce the chances of cancer when exposed to HPV. When it comes to viruses and cancer, our goal, research community's goal, is to effectively diagnose, treat, and prevent all cancers caused by viruses. That's the ultimate goal.
So infection with these viruses may, for example, papillomavirus, because papillomaviruses are DNA viruses, and DNA viruses, when enter the host cell nucleus, they are integrated into the host DNA genome. So जब ये उसका part बन जाते हैं, then they may that particular part is also expressed, और उसके फिर repercussions हो सकते हैं different. Viruses use the host cells' DNA replication machinery to replicate their own genomes, to produce a large number of infectious viral particles within a single host cell. The virus breaks through the normal constraints on DNA replication. A virus जब infect करता है किसी भी cell को, it has to produce its progenies, millions of other viruses. So what it does, it disturbs the maintenance process, it disturbs the control process of the replication. उसका काम ये है कि वायरस की और औलाद पैदा होती रहे. But because of that thing, the normal maintenance machinery or the normal control machinery is being disturbed, or is uncontrolled process की वजह से tumors भी बन सकते हैं. Sometimes the viral genome is replicated in parallel with the host cell's DNA, which can be either integrated into the host genome or as an extra chromosomal plasmid in the course of ordinary cell division cycles. Virus genome जो है ये host के genome का part भी हो सकता है या एक plasmid की सूरत में भी, क्योंकि आपने bacterial plasmids देखे that are extra from the nucleus. During this latent phase, accidents can occur that prematurely activate some of the viral proteins that the virus would normally use in its replicative phase. So some of the DNA viruses that are known to cause different kinds of cancers. For example, we have the family Papillomavirus. Papillomavirus, many distinct strains. So papillomavirus की बहुत सारी genotypes हैं. So some of the genotypes are directly linked with the emergence of cancers. Usually they cause warts. जो lesions skin पे निकल आते हैं, those are benign. But then if these warts are present in the uterine cervix, then a carcinoma may develop. इसके अलावा हिपेटिना वायरस फैमिली है, हेपेटाइटिस बी वायरस, herpes वायरस फैमिली है, फॉर एग्जांपल एप्स्टाइन-बार वायरस और ह्यूमन herpes virus 8. So these are some of the cancers that are caused by different types of DNA viruses. All these viruses are DNA viruses. They have DNA as their genetic material. And then their distribution, if we talk about the papillomavirus, they are present worldwide. Hepatitis B virus is mostly prevalent in Southeast Asia or tropical Africa. If we talk about the Epstein-Barr virus, it is in West Africa or New Guinea, South China, Greenland, Central and Southern Africa. So these viruses may in certain regions be linked with cancers, or in other regions they will cause asymptomatic disease. So they, as I said previously, कि ज़रूरी नहीं है कि ये cause करे. Maybe अगर ये वायरस पाकिस्तान में हो तो इनका कैंसर के साथ कोई लिंक नहीं होगा, but यही वायरस अगर अमेरिका में है, so it means there is a population factor as well. जो population है उसकी genetic factors भी important है. RNA viruses को देखें तो retrovirus family है, इसमें human T cell leukemia virus type 1, human immunodeficiency virus or HIV virus that may also cause a cancer type, Kaposi sarcoma. Then we have flavivirus family like the hepatitis C virus that may cause liver cancer or hepatocellular carcinoma, and the distribution is worldwide. For all these viruses, the number of people infected is much larger than the number who develop cancer. As I mentioned, it's not a must, but there is a likelihood that the viral infections may result in cancers or tumors. So let's talk about the papillomavirus. It's a DNA virus. So how the DNA viruses, they replicate in the nucleus. Once the nucleocapsid of DNA virus it enters the host cell, it proceeds to the nucleus where it mimics the genome of the host cell. जो host है उसका genome भी वही ATGC है, इस virus का genome भी वही molecules है ATGC. Both are DNA genomes. So usually the viral genome is replicated using the host cell DNA polymerase. The viruses, they don't have their own DNA polymerase. They use the polymerases of the host cell and then just become part of the replicative process. The resulting transcripts carrying information encoding viral proteins are then transported to the cytoplasm and seen as a template by the host cell ribosomes. ये वो replication का process है जिसके ज़रिए viruses अपनी proteins जो है ये बनाते हैं, और यही वजह है कि आप viruses को use करते हैं for gene therapy. We use lentiviral vectors or adenoviral vectors because these viruses, they can enter the host cell and they can insert their genome into the host genome. So what we do is we take out the pathogenic genes out of, let's say, adenoviruses or lentiviruses. We insert the corrective human genes into them. The virus will go in. It's like a missile. जैसे आपने target ले लिया वो गया उस जगह पे. And insert the corrective copy of the human gene into the human genome. तो वहाँ पे वो जो प्रोटीन जो नहीं बन रही थी या truncated versions उसके बन रहे थे वो अब ठीक से बनना शुरू हो जाएगी if everything goes right. The papilloma virus is primarily responsible for the emergence of carcinoma of the uterine cervix. They have double-stranded circular DNA chromosomes, which are about 8,000 nucleotide pairs or 8,000 base pairs. So this is the warts. The infection with papilloma virus may cause warts like this. In papilloma viruses, the protein products of the viral genes E6 and E7 bind to two key human tumor suppressor proteins. जो वायरस के ये दो जीन्स हैं, इनकी प्रोडक्ट्स जो हैं, ये इन दो होस्ट जीन्स जो कि RB and P53 है, इसके साथ बाइंड हो सकते हैं, putting them both out of action and so permitting the cell to replicate its DNA and divide in an uncontrolled way. इन दोनों के बारे में हमने पढ़ा, they are the tumor suppressor genes, they control the cell growth, but the viruses through these two proteins can deactivate their function. अब यहाँ पे एक और जो आजकल रिसर्च हो रही है कि वायरस गया, उसकी कुछ प्रोटीन्स हैं जो कि नॉन ह्यूमन प्रोटीन्स के साथ अटैच होती है. So you use analogs. What they do is they use the protein mimics. वो उसमें ऐसी प्रोटीन डाल देते हैं जो कि होती ह्यूमन की तरह है, but it's not actually human. So the virus proteins will bind to those proteins and not the actual human host protein. तो वो उसके साथ बाइंड कर जाती हैं और मे भी फिर आगे चलकर उनका कोई इनहिबिटरी इफेक्ट होगा. So these kind of analogs are being actively used. ऐसे बन रहे हैं, ऐसी एंटीवायरल्स हैं जो कि इस तरह से काम करती हैं. So viral proteins, they mediate the controlled replication of the virus in outer cell layer. This is the host chromosome. This is the chromosome of the papilloma virus. The viral chromosomes are stably maintained in the basal cells of the epithelium as plasmids, whose replication is regulated so as to keep step with the chromosome of the host. So this is one case, like the virus genome is held or is it holds like a normal plasmid. यहाँ पे ये अलग से होगा, अभी पार्ट नहीं है होस्ट क्रोमोसोम का. इट बिहेव्स इंडिपेंडेंटली, ये मौजूद है न्यूक्लियस में या सेल में और अपना काम कर रहा है. But sometimes rare incidents can cause or accidents can cause the integration of a fragment of such a plasmid into chromosome of the host, altering the environment of the viral genes in the basal cells where the stem cells of the epithelium reside. This can disrupt the normal control of viral gene expression. And the unregulated production of certain viral proteins, the prime proteins are E6 and E7, that interferes with the control of cell division by binding to P53 or RB proteins. ये वो अंडरलाइन मैकेनिज्म हैं ब्रॉडली, जो वैसे तो ये काफ़ी कॉम्प्लिकेटेड होते हैं अगर आप स्पेसिफिकली कोई पेपर पढ़ें इसके बारे में, बट these are just the summary information about how this whole thing unfolds. So we have the HPV vaccine. वेस्ट में ये वैक्सीन काफ़ी कॉमन है. इट्स अ प्रोफिलैक्टिक मेजर. प्रोफिलैक्टिक मेजर मीन्स कि डिजीज के ऑनसेट से पहले ही आपने ऐसे मेजर्स अडॉप्ट किए कि वो इन्फेक्शन अगर हो भी जाए तो वो डिजीज ना हो. थेराप्यूटिक मेजर, ये डिजीज हो गई और उसके बाद आपने, सो वैक्सीन्स आर टेकन in both the instances. They can be prophylactically administered or they may also be therapeutically administered. अब ये डिपेंड करता है. अगर आपके पास एक सिस्टमैटिक वे है, पब्लिक हेल्थ सिस्टम है, तो वो प्रोफिलैक्टिकली जो भी सर्विकल कैंसर्स तो है, they are more common in young population, so they will vaccinate their young population even before the infection comes in. This is a prophylactic measure. So we have the vaccine. We have the HPV vaccines, which can reduce the incidence of the cancer, let's say, or the infections of the human papilloma virus. What vaccines does? It trains your immune system. ये आपके immune system को train करती है if the infection occurs, if the real infection occurs, तो जल्दी से जाके उसे kill कर दे. ठीक? But sometimes, like if a new virus comes in, like when SARS-CoV-2 came in, it's a new virus, it's a coronavirus family, it belongs to the coronavirus family, but sometimes it results in the hyperactivation of the immune system, and that's why what we say is multi-organ failure happened. आपने डॉक्टर्स की रिपोर्ट्स देखी होगी मल्टी ऑर्गन फेलियर विद अननोन कॉजेस. इट्स मोस्टली बिकॉज ऑफ योर इम्यून सिस्टम. कोई नया वायरस आ गया, कोई नया पैथोजन आ गया और इम्यून सिस्टम उसे जानता नहीं है, डजंट रिकॉग्नाइज इट. सो व्हाट इट डज, इट जस्ट स्प्रेड्स द न्यूज़ टू द होल सिस्टम कि भाई सबको जाके आपके पास जो भी असलहा है आप इस पे मारे, इसको खत्म कर दें. बट ड्यूरिंग दिस प्रोसेस ऑफ this battle between the pathogens and the immune system, जो आपके cells होते हैं वो they get destroyed. अब वायरस जहां जहां जा रहा है immune system उसको किल करने शुरू कर देता है. It goes to the heart cells are damaged. The immune system actually wants to kill the virus, but then the virus is inside the cells, so it just solely kill the cells. वो liver cells में जा रहा है, वहां भी killing हो गई. तो बहुत सारे SARS-CoV-2 के ऐसे केसेस आए जो एक दिन में ही मर गए. I mean, they got infected one evening. Certain my relatives as well. We had a lady in our family जिनको एक शाम निमोनिया हुआ और सुबह वो she expired because of the multi-organ failure. I read about a case जहाँ पे एक लड़का जो था वो आ गया, ये मरी वगैरह गया था sometime, and he came back. And suddenly he developed a fever of 107. 107 degree का fever उसने develop किया. Fever is an immune response. ये आपकी body का एक response होता है किसी भी infection को control करने के लिए. But sometimes this response gets out of control as well. And when that 107 happened, his brain collapsed. वो बच तो गया लेकिन वो सारी जिंदगी के लिए बिल्कुल brain damage हो गई. The brain mostly cannot recover. So sometimes your immune system gets out of control as well. So vaccines can stop both. They can train your immune system against that novel pathogen. So this is a very interesting audio. ये मैंने BBC पे सुनी थी. It's a very interesting audio. Professor Sir Anthony Epstein, जिन्होंने एप्स्टाइन बा वायरस जो है वो डिस्कवर किया था. ये जब हम कोर्सेज लेते हैं तो उसमें भी मैं ये प्ले करता हूँ. So more than 50 years ago, a team of researchers in London introduced the world to an extraordinary new virus now called Epstein-Barr virus, EBV. Sir Anthony Epstein and Dr. जो इनके postdoctoral student थे, Yvonne Barr, इन दोनों ने मिलकर ये डिस्कवरी की थी और इसी वजह से इस वायरस को एप्स्टाइन-बार वायरस भी कहते हैं. About 95% of us have been infected by the virus. यानी कहीं ना कहीं हमें ये इन्फेक्शन हुआ है. 95 is a big number. And we stay perfectly healthy. So nothing happens. We even don't know that we are infected with the virus. But sometimes, under certain circumstances, it can cause a range of seemingly unrelated diseases. For example, Burkitt's lymphoma. ये टाइप है कैंसर की जो कि अफ्रीकन चिल्ड्रन में होती है if they are infected with the Epstein-Barr virus or EBV virus. To nasal cancers in southern China, या nasal cancers भी हो सकते हैं southern China में, and glandular fever in Europe and the US. So in different regions, this infection sometimes cause different types of disease phenotypes. Southern China में अगर इन्फेक्शन हो रहा है तो फिर नेजल कैंसर्स का चांस ज्यादा है. अगर अफ्रीका में हो रहा है देन बर्किट्स लिम्फोमा का चांस ज्यादा है. और अगर यूरोप और यूएस में इसके इन्फेक्शन हो सो देयर मे बी चांसेस ऑफ डेवलपिंग ग्लैंडुलर फीवर. So in this audio, ये एक ऑडियो है, Sir Anthony Epstein, he spoke with Dr. Graham Easton how he discovered the virus. ये बड़ी इंटरेस्टिंग टॉक है, आप सुने. इसमें बड़े रिसर्च जो आइडियाज हैं ये भी इन्होंने डिस्कस किए.
50 years ago, almost to the day, a team of researchers in London introduced the world to an extraordinary new virus, now called Epstein-Barr virus, or EBV, after its discoverers, Sir Anthony Epstein and Dr. Yvonne Barr. About 95% of us have been infected by the virus and we stay perfectly healthy. But in some people, under some circumstances, it can cause a range of seemingly unrelated diseases, from Burkitt's lymphoma in African children to nasal cancers in South China and glandular fever in Europe and the US. Sir Anthony Epstein told me how it all started in London in 1961, when he heard an unknown surgeon from Uganda giving a lecture about a bizarre new children's cancer which caused jaw swellings. It was subsequently named after him, Burkitt's lymphoma. But it was when Burkitt started talking about the geographical spread of the tumour that the penny dropped for Epstein.
That was the thing that set me hopping up and down in my seat after his first 20 minutes. Its distribution appeared, from his very preliminary data, to be dependent on temperature and rainfall. He didn't seem to see it where there wasn't enough rainfall and where the temperature dropped. And this, of course, suggested to me immediately that there must be some biological agent involved. And I had been working for several years on the then unfashionable topic of chicken viruses which cause cancer. I therefore had virus-inducing tumours very much in the front of my head. And after he'd been talking for a few moments, I immediately thought, this is a tumour virus.
ये स्टोरी शुरू हुई. He attended a lecture जो कि एक डॉक्टर दे रहा था. We attend many lectures. We have seminars here in iccbs जहाँ हम randomly उन lectures को attend करते हैं. So he didn't know about this virus. और ये डॉक्टर साहब जो थे, ये एक न्यू डिजीज के बारे में बता रहे थे जो कि जॉ स्वेलिंग, वो आपके जॉज होते हैं, इसमें स्वेलिंग जो है वो कॉज करती है. और ड्यूरिंग द प्रेजेंटेशन ही गेव सम डिस्ट्रीब्यूशन डेटा, एनवायरमेंटल डेटा. और वो डेटा क्या था कि जहां पे बारिश ज्यादा हो रही है या टेम्परेचर गिर रहा है, समहाउ वहां पे इंफेक्शंस भी बढ़ रहे हैं. And this guy, like Anthony, Sir Anthony, because he was working with chicken viruses, तो ये एक common indicator है कि अगर आपकी environmental factors के साथ कोई disease change हो रही है, उसका incidence change हो रहा है, then it means the disease is moving actually. जैसे डेंगू है फॉर एग्जाम्पल, जब बारिश ज्यादा होती है तो डिजीज भी ज्यादा होती है. क्यों ज्यादा होती है? बिकॉज द पॉपुलेशन ऑफ द वेक्टर इंक्रीजेस. तो इस डेटा से उस डॉक्टर को तो ये पता नहीं चला. He just presented some data, but this guy, he knew that there is some biological agent, there is some vector that is carrying this pathogen from one place to another. तो जब आप भी मतलब कोई सेमिनार अटेंड करते हैं, so just focus on what the presenter is saying. Maybe you are working on something और वो कुछ और कॉन्टेक्स्ट में कह रहा हो, but that becomes highly relevant to what you are doing.
Cancer virus being carried by some insect vector or some tick. And that was why it was temperature dependent.
And that was the beginning of a very fruitful relationship, wasn't it, between you and Dennis
Well,
Burkitt?
I immediately decided I was stopping all my work and would concentrate on finding a virus in this weird tumour.
Sir Anthony then visited Uganda to arrange a supply route for samples of lymphoma in which to start the search for the virus. But success didn't come easily.
The trouble was that every sample that we examined by the then current virological methods for diagnosis proved negative. There was nothing. And we even looked in the electron microscope, which was very unusual at that time. And I was extremely disappointed that this new tool gave nothing.
That must have been very scary at the beginning of your career.
What they did, he went to Uganda, जहाँ पे ये disease थी. He took the samples from the patients, he brought them back to the laboratory and tried to see what the virus is, how it looked. But the current methods, the prevalent methods, in those times, they couldn't diagnose the existence of the virus. You couldn't find anything. जब पता तो उसे था कि there is a virus present in the cells that is actually causing them to develop tumors and or cancers or Burkitt's lymphoma, but he couldn't diagnose it. So for you, like when you are writing a paper, you need to have some results. When you are trying to link a pathogen with a particular disease, तो ये तो एक common principle है जो कि Koch postulates आपने पढ़े हैं. वो 6 postulates हैं. क्या आपने एक sample दिया, उसमें वो pathogen आपने देखा, you found it through different methods, and that sample then developed into a disease. It means the pathogen is linked with the disease, जो कि normal cell population में नहीं होता. But Sir Anthony couldn't find the viruses in the samples. Why? He will discuss.
On for 2 years, really, with
For 2
there was
years,
no employment legislation in those days. I actually had no letter of appointment, no terms and conditions of service, and didn't know from 1 year to the next whether the head of department would reappoint me.
It was a frustrating 2 years, but Epstein wondered if the viruses could be lying dormant and might only be activated if the malignant cells themselves were grown in the laboratory. But at the time, no one had managed to grow human lymphocytes in the lab.
Then there was an amazing stroke of luck. One of the flights bringing in our samples from Uganda was delayed by fog and diverted to Manchester. And it didn't come to us in London until late on a Friday afternoon, actually the 5th of December 1963. The fluid in which the sample was suspended was cloudy. Well, this suggested that somehow it had got contaminated on the way and that it was full of multiplying bacteria and therefore useless for investigation. But before we threw it away, packed up and went home for the weekend, I looked at it under a wet preparation with a light microscope, and instead of seeing large numbers of bacteria, I saw huge numbers of free-floating, healthy-looking tumour cells, which had got shaken free from the cut edges of the sample on the long journey.
It was a lucky break. This reminded Epstein that mouse lymphomas would only grow in this sort of suspension of free-floating single cells, rather than the usual glass surfaces for support. He tried it on the human cells, and they grew. It's now the standard technique across the world for growing these sorts of cells. But the usual biological tests for identifying existing viruses just didn't work. So he had to use a new device to hunt the virus down.
It was extremely biologically inert. So I looked at it in the electron microscope. That was not a way of demonstrating viruses in those days. That was completely unorthodox. And the very first grid square that I looked at in the machine had a cell with virus particles. Well, I was first of all exhilarated, but then I was extremely frightened in case the electron beam burnt the specimen up and I would never see it again. So I switched off and I walked around the block in the snow. It was February and I...
समझ आया आपने क्यों उसने तो जो कल कर रहे हैं कल्चरिंग आप में से कोई सेल कल्चरिंग करते हैं आप करते हैं क्या हुआ आपने तो खैर सुनी भी है समझाए कि क्या हुआ
स्टार्ट में इन्होंने जो वहाँ से सैंपल लिए थे तो उनको इनोकुलेट किया जिसमें वो टर्बिडिटी है वैसे वायरल में नहीं आती बैक्टीरियल में आती है तो स्टार्ट में ये कह रहे थे कि बैक्टीरियल पॉपुलेशन काफ़ी ज़्यादा है इसमें एंड देन फर्दर जो चीज़ें डिस्कस हुई हैं वो तो सबने
आप तो टोटली गलत बात कह रहे हैं
मैंने स्टार्ट में इसलिए पिक की थी
अच्छा पहले जो वो देख रहे थे because he was trying to see whether the virus is in there and he couldn't find it. He knew that the virus is in there, but what was happening? जो samples आ रहे थे जो cells के they were mostly static cultures. अब जो cell culturing के साथ काम करते हैं उन्हें पता है कि static culture क्या होता है जहाँ आप cells को grow करते हैं petri dish. Adherent cells. What happened is one of the flight got delayed. Whatever reasons, वो cell गोदाम में पड़े रहे and during the flight they were like shaking. वो जो शेक हो रहा था उस वजह से वो कैंसर सेल्स ग्रो होने लगते हैं। अब ये बड़ी नॉर्मल सी बात है सेल कल्चरिंग में कि जो भी सेल्स आप ग्रो करना चाहते हैं वो आइदर स्टैटिक कल्चर्स में ग्रो होंगे या शेकिंग कल्चर्स में ग्रो होंगे। आप इंक्यूबेटर में शेक करते रहते हैं, अगर आप उनको स्टैटिकली ग्रो करने की कोशिश करेंगे वो कभी भी ग्रो नहीं होंगे। So that was the discovery. Like he was trying to discover the virus, but he discovered the shaking cultures as well, जिनको हम suspension cultures कहते हैं। ये फर्स्ट डिस्कवरी थी। इससे पहले उसने कहा भी कि हम कैंसर सेल्स को ग्रो नहीं कर सकते। तो जब वो सस्पेंशन कल्चर्स में ग्रो हुए तो उस वक्त उसे इलेक्ट्रॉन माइक्रोस्कोप में बड़े ही खूबसूरत अंदाज से वायरस पार्टिकल्स नजर आने लग गए। स्टैटिक कल्चर्स में वो नजर नहीं आ रहे। This was a discovery line. He discovered the virus and he also discovered the suspension cultures.
And no coat on. And when I came back calmer, I recorded what I had seen and I recognized it at once as a member of the herpes family of viruses. But which herpes virus was impossible to tell. There were about 5 human herpes viruses known then. There are now 9. But the curious thing was that any of the known ones then would have wiped the culture out as they were replicating, and this was not happening. So I really had the feeling that this was some...
Then he discussed further that these viruses are present in the samples. He couldn't characterize them, so fully characterize इन्होंने नहीं किए थे, but what he wrote in the paper, जब इसने पेपर लिखा कि these viruses need to be characterized, should be characterized. और इसके बाद WHO ने एक बड़ी स्टडी शुरू की जिसमें thousands of patients को स्क्रीन किया गया, उन्हें ये वायरस मिला और उसके बाद फिर इसका नाम जो है एप्स्टाइन-बार वायरस रखा गया, and it is linked to the cases of Burkitt's lymphoma in African countries. But the thing is like if this virus infection may culminate into different other infections. So it depends on the host immune system. Further audio सुनेंगे तो उसमें ये immune system का जिक्र करेगा। जो मसला था वो ये था कि बर्किट्स लिम्फोमा उन बच्चों में डेवलप हो रहा था जिनको मलेरियल इन्फेक्शन हो जाता था। वायरस से भी इन्फेक्शन हुआ और फिर मलेरियल इन्फेक्शन भी हो जाता था। Because of the malarial infection, somehow their immune cells got altered. आपको पता है मलेरियल इन्फेक्शन में मलेरिया पैरासाइट जो है वो इम्यून सेल्स को इन्फेक्ट करता है आमतौर पे। तो उसकी वजह से जो भी चेंजेस आती थी, and because of those changes, this infection was turning into a Burkitt's lymphoma case. So you need to study कि comorbidities कौन सी हैं। एक पेशेंट है उसमें कैंसर डेवलप हुआ है, पहले इन्फेक्शन हुआ, then what was the secondary infection, what was the comorbidity status, whether the patient is a heart patient or a diabetic patient. ये सारी clinical information here examine की जाती है, and then you will have to develop the linkage. So इस particular case में इन्होंने 2 discoveries की. This is how the whole science happens. You are working on something else and something else happens. आपने antibiotics का case तो सुना ही होगा. He was working on a fungi and one day he found out that in a petri dish the bacterial population was killed. वो काम ही उस पे नहीं कर रहे थे. He just and then thought, okay, let's see what's in there, and he discovered the antibiotics. इसको कहते हैं serendipitous discoveries. साइंस में 90% डिस्कवरीज जो है वो serendipitous होती हैं. मींस अचानक से कोई चीज आपने डिस्कवर कर ली, आप उस चीज पे काम नहीं कर रहे थे, बट somehow that happened. और अभी तक 90-95% जितनी भी डिस्कवरीज हुई हैं वो ऐसे ही हुई.
इन्होंने एनवायरमेंटल सैंपल्स को नहीं चेक किया, क्योंकि जो इन्होंने पहले जो सेमिनार जैसे अटेंड किया था उसमें था कि जहाँ पे रेन ज़्यादा हो रही है और उस तरह का है,
वो बच्चों से ही सैंपल लिए थे ना. जो डॉक्टर वो प्रेजेंट कर रहा था तो उसने बच्चों से, अब बच्चों में एक ऐसी डिजीज आ गई जो नोन नहीं थी, जॉ स्वेलिंग. मींस उनके जॉज में स्वेलिंग हो रही थी. उन्होंने उससे सैंपल लिए. मे बी दे टुक अ बायोप्सी, जॉ से बायोप्सी ली, लेकिन उन्हें कुछ मिल नहीं रहा था. बट ड्यूरिंग दैट डिस्कशन उस डॉक्टर ने ये भी बताया कि ये डिजीज जो है ये डिफरेंट इन्वायरमेंटल फैक्टर्स जो है इसको अफेक्ट कर रही है, भी अननोन डिजीज है, इसके बारे में हमें कुछ नहीं पता। जैसे पैंडेमिक आपके पास आया तो ये सारी चीजें डिस्कस हो रही थी कि अभी वायरस के बारे में ज्यादा पता नहीं था, लेकिन ये मुख्तलिफ इलाकों में कैसे फैल रहा है, मुख्तलिफ टेम्परेचर्स पे कैसे फैल रहा है, मुख्तलिफ जेंडर ग्रुप्स पे कैसे फैल रहा है। So the researchers were discussing all these things. They were working. These are the factors that need to be considered. ये सारे फैक्टर्स आपने कंसिडर करने होते हैं। अब हो क्या रहा था कि इस केस में बर्केट्स लिम्फोमा के केस में मैंने पहले भी डिस्कस किया कि जहां पे रेन और टेम्परेचर चेंज हो रहा था वहां पे ये इसका इंसिडेंस ज्यादा था या कम था, जो भी था, ठीक है। अब रेन और टेम्परेचर वायरस पे तो कोई इफेक्ट उस तरह नहीं करता, बट समथिंग वाज कैरिंग दैट वायरस टू डिफरेंट लोकेशंस। और चूंकि ये साहब जो थे ये ऑलरेडी चिकन वायरसेस पे काम कर रहे थे, वेक्टर बॉर्न वायरसेस पे काम कर रहे थे, तो इनके जहन में फौरन ही आ गया कि ये कोई टिक होगी या मॉस्किटो होगा जो कि इस वायरस को एक एरिया से दूसरे एरिया में लेकर जा रहा। So now we move on to other cancer causing viruses. For example, we have the hepatitis B and C viruses. Mostly they cause chronic inflammation. आमतौर पे जो हेपेटाइटिस बी और सी वायरसेस है ये इन्फ्लेमेशन कॉज करते हैं, जिसको हम हेपेटाइटिस कहते हैं। क्या होती है इन्फ्लेमेशन? हेपेटाइटिस कॉज करते हैं इन्फ्लेमेशन। इन्फ्लेमेशन क्या है? हाउ वी डिफाइन द इन्फ्लेमेशन? जी हाँ, रेडनेस तो वो है ना, उसकी एक सिम्टम है, ठीक है। But what is, how we define inflammation?
Inflammatory molecule रिलीज होते हैं सर। बेसिकली जो हमारे पास फॉस्फोलिपिड बाइलेयर्स है वो एक एराकिडोनिक एसिड्स में कन्वर्ट होते हैं।
अब आप उसका अंडरलाइन मैकेनिज्म बता रहे हैं। Let's say in one line, if I ask you what inflammation is, clinical information दे आप उसकी प्रोटीन केमिस्ट्री में चले गए हैं कि ये मॉलिक्यूल होता है, ये नहीं होता, बट अब एक आम बंदा आपसे पूछे इन्फ्लेमेशन क्या चीज है, भाई मुझे समझाएं, वन लाइन में समझाएं।
अनयूजुअल इंक्रीज साइज
जी,
जो नॉर्मल साइज है उसका अनयूजुअल हो जाना,
ये भी एक सिम्टम हो गया, ठीक है। रेडनेस जिसे हम कहते हैं, ठीक है, वो एरिया बिल्कुल सुरख हो जाता है, वो भी अगेन इट्स अ सिम्टम। जी, इन्फ्लेमेटरी मार्कर्स, ठीक है, क्या है मतलब? फिर वही बात ना, इन्फ्लेमेशन हो गई ना, allergic reaction. Yeah, it's an allergic reaction. Simple. It's an allergic reaction. Inflammation is an allergic. ये एलर्जी है जहाँ पे आपका इम्यून सिस्टम कुछ गड़बड़ करता है जिसकी वजह से कोई डिजीज मैनिफेस्टेशन। तो वन लाइन में मेरे ख्याल में इसकी जो डेफिनेशन है, इट्स एन एलर्जिक रिएक्शन, दैट्स इट। एंड द अदर पर्सन विल नो नाउ द इम्यून सिस्टम इज इन्वॉल्व। और ये जो वर्ड आता है टाइटिस, इस तरह की मायोकार्डाइटिस, and you will see other terminologies as well, they are all linked to inflammatory reactions. या आपको किसी मच्छर ने काटा है, टिक ने, वो एरिया रेड हो गया, that's the inflammation because the immune cells they accumulate at that place that may result in different manifestations. So normally अगर hepatitis B और C का infection हो तो chronic inflammation हो जाए for long term, tissue damage हो गया, which stimulates an extensive cell division in the liver that promotes the eventual evolution of tumor cells. और जब टिश्यू डैमेज हो जाती है, सेल्स डैमेज हो जाते हैं, मे बी इन लिवर, तो वहां पे दोबारा से सेल डिवीजन शुरू हो जाता है और समटाइम्स इट गेट्स आउट ऑफ कंट्रोल और ट्यूमर्स भी डेवलप हो सकते हैं। नॉर्मली वो क्योर हो जाएगा। In AIDS, for example, HIV infections, human immunodeficiency virus, it promotes the development of an otherwise rare cancer called Kaposi sarcoma. HIV जो वायरस है, ये मुख्तलिफ डिजीज मैनिफेस्टेशंस कॉज करता है, ये इम्यून सेल्स को इन्फेक्ट करता है, ये इसका जो घर है वो इम्यून सेल्स में है। CD4 lymphocytes, CD4 positive T lymphocytes, I guess primarily it targets these cells. और जब वो इनको किल कर देता है तो बॉडी का इम्यून रिस्पांस काफी लो हो जाता है, इट सप्रेसेस द इम्यून रिस्पांस, जिसकी वजह से पेशेंट को बहुत सारे इन्फेक्शंस हो सकते हैं। और चूंकि अब इम्यून सिस्टम जो आपका वो पूरी तरह से काम नहीं कर रहा, तो वो नॉर्मल इन्फेक्शंस भी किसी पेशेंट के लिए लीथल साबित हो सकते हैं। तो उसी सीरीज में ये एक कैंसर भी कॉज करता है, जिसको कापोजी सार्कोमा यानी कैंसर ऑफ द बोन, ठीक है, बोन कैंसर जो है ये कॉज कर सकता है बाय डिस्ट्रॉइंग द इम्यून सिस्टम, thereby permitting a secondary infection with a human herpes virus that has a direct carcinogenic action. So basically ये कैंसर कॉज नहीं कर रहा, ये क्या कर रहा है, इम्यून सिस्टम को डिस्ट्रॉय कर रहा है, और जब इम्यून सिस्टम डिस्ट्रॉय हो जाता है तो एक और इन्फेक्शन हो सकता है जो कि HHV8 वायरस से, और ये वायरस जो है फिर जाके कैंसर कॉज कर सकता है। So it's a double infection, but the primary cause, the primary reason is the infection with the HIV virus. अगर आप इसको somehow रोक दें तो फिर जो secondary infection है आपके पास HHV का, वो नहीं होगा। अब हमने वायरसेस के बारे में पढ़ा, एप्स्टाइन-बार वायरस, HIV virus. There are certain bacterial cell lines as well that can cause cancers. Chronic infection of the stomach with the bacterium Helicobacter pylori, ये बड़ा मशहूर बैक्टीरियम है, जो लोग काम कर रहे हैं वो इसको अच्छी तरह से जानते हैं. It can cause ulcers and then it appears to be a major cause of stomach cancers. ये अल्सर्स कॉज करता है और कभी कभार ये स्टमक कैंसर्स भी कॉज कर सकता है.
So Helicobacter pylori infects about 50% of people in the world. It's a gastric. So Helicobacter pylori infects about 50% of people in the world. It's a gastric pathogen that infects the stomach. It's a bacteria. In 1984, the first report of this was in The Lancet, described by Marshall and Warren. They won the Nobel Prize for this in 2005 for the discovery that H. pylori causes inflammation and peptic ulcer disease. But it also turns out that Helicobacter pylori, or H. pylori, is the most important factor in the development of gastric adenocarcinoma. So gastric cancer is the third leading cause of cancer death worldwide, and it's over 700,000 deaths per year. And Helicobacter pylori, you would think it would be very straightforward. If we know that there's a pathogen that leads to this, why can't we just get rid of it in everyone? So there's a lot of problems with that approach. First of all, there may be some actual benefits of this pathogen because it started out probably as a commensal organism. There's evidence that it goes back hundreds of thousands of years, and there's ideas that it co-evolved with humans, and that it's only when a lot of work that our group has done, not really my lab, but people that I work with, has shown that when there's a disconnect between the human genetics and the bacterial genetics, this mismatch may be one of the factors that leads to cancer, which we're studying. So there may be many people that they don't really need to be eradicated. And in fact, there's evidence that overzealous eradication may have a role in leading to autoimmune disorders, things like asthma. So we want to understand the whole process of the inflammation and how that leads to cancer.
तो ये डॉक्टर हैं, ये कह रहे हैं कि आमतौर पे जो ये H. pylori के इन्फेक्शंस हैं, ये एक commensal है. This bacteria is living with humans for thousands of years. There is a commensal relationship. We दोनों ही host and the pathogen, they live happily, but sometimes something happens. कोई इन्फ्लेमेशन, कोई एलर्जिक रिएक्शन ऐसा हो जाता है जिसकी वजह से यही बैक्टीरिया, जो कि आमतौर पे हमारे लिए फायदेमंद है, ये ट्यूमर्स और कैंसर्स या डिफरेंट अदर डिजीज कंडीशंस कॉज करता है. तो इसकी बायोलॉजिकल साइकिल को पूरी तरह से पढ़ना भी चाहिए, कैरेक्टराइज भी करना चाहिए, ताकि वो क्या फैक्टर्स हैं जिनकी वजह से एक कमेंसल बैक्टीरिया भी पैथोजेनिक बैक्टीरिया में कन्वर्ट हो जाता है. तो ये जितनी भी वीडियोस हैं इसमें आप लोग देख रहे हैं, दे आर द लीडिंग रिसर्चर्स. मे बी someone of you is working in cancers, they may contact these guys. अगर आप जिन फील्ड्स में ये काम कर रहे हैं, अगर आप उसी फील्ड में काम करना चाहते हैं, like this guy is working with H. pylori and you are also interested in working with H. pylori and stomach cancers, you may contact him. Ask him about whether if you are looking for a PhD, you are looking for a postdoc, or maybe you are looking for a collaboration, you may contact these guys. And इसी तरह से लोग जो है PhD positions या postdoc positions इसी तरह से find. So now we will move into the challenges in the search for cancer cure. Okay, we will stop here and we'll discuss that tomorrow. Okay, this is the last part of the lecture. Okay, maybe it takes more than 1 hour to fill 12:30 se zyada. So if you have any questions. In lectures का आपको कोई फायदा भी हुआ है? ठीक है, there are a lot of lectures here in this. Whether they benefit you from there. Otherwise, that's just the information. ये information आपको कहीं से भी मिल सकती है. These are the videos that are available on the internet. But whether it benefits.
Yes.